to our channel Bioholic with your Friday educator Olivia. In the part 7 of photosynthesis, today I will describe about an important thing, that is the photorespiration. And after the class is over, don't forget to check the description box to attend the self-assessment test of today's class. Okay, so let's start. In previous parts, we have learned the C3 or Kelvin cycle, where we learned about the Rubisco enzyme that shows carboxylase activity. But the full form of Rubisco is RUBP, that is ribulose 1,5-bisphosphate carboxylase oxygenase. So it has both carboxylase and oxygenase activity. In the C3 cycle, it acts as a carboxylase enzyme to fix carbon dioxide to RUBP and make glucose. But sometimes it also shows oxygenase activity, but that is very harmful for the plants. Today we will learn what happens when Rubisco shows oxygenase activity. In normal condition, in exposure to sunlight, in the daytime, the stomata of the leaves are open, and by which water and oxygen come out from the leaf, and carbon dioxide from the environment enters the cell. But during scorching heat of the sun, when the temperature is too high, then if the stomata remains open, then plants will lose all of its water. So the plants may also die. That's why plants then close its stomata. But now another problem rises. Now oxygen cannot come out and carbon dioxide cannot enter the cell. So the concentration of the carbon dioxide decreases inside the cell, and the oxygen, that is the byproduct of the light reaction, starts accumulating in the plant cells. So in this condition, that is in exposure to high temperature and high oxygen concentration inside the cell, plants, mainly the C3 plants, shows photorespiration. So what is photorespiration? Photorespiration refers to a process where the enzyme Rubisco oxygenates RUBP, wasting some of the energy produced by photosynthesis. This process reduces the efficiency of photosynthesis. So when the oxygen availability is high in the cell, then Rubisco shows an affinity towards the oxygen and not towards the carbon dioxide, because then the carbon dioxide concentration is low. Then Rubisco starts to show its oxygenase activity. So the conditions of photorespirations are altered substrate availability, or the lowered carbon dioxide or increased oxygen, and increased temperature. Okay? Now the photorespiration involves 3 organelles, that is the chloroplast, paroxysm, and mitochondria of the plants. And in this process, oxygen is taken and carbon dioxide is excreted out. That means it has similarities with our respiration process, in which we take oxygen and release carbon dioxide. And this process occurs in the scorching heat of summer, which means in exposure to light. That's why this process is known as photorespiration. Clear? This is the full photorespiration process, where chloroplast, paroxysm, and mitochondria are involved. So at first, RUBP combines with oxygen, unlike carbon dioxide in the C3 cycle. After this combination, phosphoglycolic acid is formed, which is the first stable product of the photorespiration. And this phosphoglycolic acid has 2 carbon in its structure. That's why sometimes the photorespiration process is called as C2 cycle. Okay? Also, there 1 molecule of phosphoglyceric acid is formed in this reaction. After that, it is converted into glycolic acid that enters in the paroxysm. And in the paroxysm, the glycolic acid is converted into glyoxylic acid and hydrogen peroxide. and later it is converted into glycine, and this glycine enters the mitochondria, and then the glycine, two molecules of glycine, is converted into serine, and in this reaction carbon dioxide is released. We know to produce glucose we need to fix carbon dioxide, but in this case carbon dioxide is being released, so obviously glucose cannot be made. That's why the photorespiration is actually an unnecessary problem of the C3 plants. Nextly, the serine enters the paroxysm and later converted into glyceric acid, which is then enters the chloroplast. Here the glyceric acid is converted into phosphoglyceric acid, but here again it is wasting another thing, that is the ATP. It is using one molecule of ATP, but there is no fruitful effect as it is regenerating the RUBP without producing any glucose or any carbohydrate molecule. So here we can see that no glucose is formed, moreover ATP is used, thus there is waste of energy and also there is loss of carbon atoms. This is the simplified comparison between the photorespiration and Kelvin cycle. In photorespiration, two molecules of phosphoglycolate and one molecule of 3-phosphoglycerate is produced. And because the phosphoglycolate has two carbon atoms, that's why this cycle is also called as C2 cycle. But in case of Kelvin cycle, two molecules of 3-phosphoglycerate is produced and the phosphoglyceric acid or 3-PGA has 3 carbons in its structure. That's why it is called as C3 cycle. And in the Kelvin cycle, we know that glucose is made, but in photorespiration, glucose molecules cannot be produced. So photorespiration is definitely not a win from a carbon fixation standpoint. However, it may have other benefits for the plants. Those are that under condition of intense light and poor availability of carbon dioxide, photorespiration plays defensive role. With insufficient availability of carbon dioxide and light energy cannot be fully utilized, and excess light can cause photooxidation and damage the plant. Photorespiration provides protection against this, and in this process, in C3 plants, oxygen binds to rubisco and so carbon dioxide fixation is decreased. But in C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of carbon dioxide at the enzyme site. So to avoid this problem, C4 plants are evolved and they shows a significant mechanism by which oxygen cannot bind to the RUBP. That's why there is no photorespiration and thus no hampered photosynthesis. What are those mechanisms? Next Friday we will learn about the C4 plants and C4 pathways. So today's class was all about photorespiration process. If you have any doubts or any queries, contact through our mail ID or you can also comment below. And if you are new here, don't forget to subscribe our channel to get all the free classes and self-assessment tests, mock tests, study materials absolutely free. So I'm signing off now. See you in the next Friday. Till then, stay tuned with Bioholic.